Biofertilizers and medicinal plants. Biofertilizers: Rhizobium, blue-green algae. Medicinal plants: Adhatoda, zeylanica, medic, Pasaka; Aloe vera, lin, Aloe; Ocimum sanctum, lin; Asparagus racemosus, lin, Asparagus; Azadirachta indica, Jus, Margosa. Mushroom cultivation. Fertilizers are the substances which improve the fertility of the soil and help plants grow and synthesize more food, giving high yield and better quality of food crops. Fertilizers are classified into chemical fertilizers, biological fertilizers, and biofertilizers. Biofertilizers: the microorganisms which increase the soil fertility by fixing atmospheric nitrogen, adding it to the soil, are called biofertilizers. They are called diazotropes since they fix atmospheric diatomic nitrogen. Nitrogen, under the presence of microorganism nitrogenase, gives NH3 ammonia, which is nitrogen fixation. Biofertilizers are symbiotic and non-symbiotic. Symbiotic: these are the microorganisms which fix atmospheric nitrogen in association with the plants. Symbiotic biofertilizers include certain bacteria like rhizobium and blue-green algae like Anabaena nostoc. Non-symbiotic biofertilizers are the microorganisms which fix atmospheric nitrogen without associating with the plants. Non-symbiotic biofertilizers include certain bacteria like Azotobacter, Chlorobium, Clostridium, etc. Rhizobium: it is a gram-negative bacilli that lives as an endosymbiont. Rhizobium may occur freely in the soil but fails to fix nitrogen in the free state. It is aerobic, non-motile, and non-spore-forming bacterium. It is a nodule-forming bacterium. The nodule-forming and nitrogen-fixing activity of the rhizobium is host-specific. Formation of a nodule is under the control of NOD gene, nodulin gene of the host. Affinity of rhizobium to the particular leguminous plant is due to the proteins called lectins or lecithins present in the roots. Phytoagglutinins, lecithins, phospholipid, serve as recognition chemical substances between the two symbionts, that is, the plant and the bacteria. Rhizobium fixes nitrogen in the presence of red pigment called leg hemoglobin. The nitrogen fixation by rhizobium is catalyzed by the bacterial enzyme nitrogenase. The production of this enzyme in the rhizobium is controlled by the nitrogen-fixing gene NIF gene. The diagram showing root system with nodules and TS of a root nodule is shown. Blue-green algae. They are presently referred to as cyanobacteria. They are prokaryotic, colonial or free-living, filamentous forms mostly found in moist soils or paddy fields. Many members of cyanobacteria are diazotropic. A diazotroph is an organism that is able to grow without external sources of fixed nitrogen. BGA have natural nitrogen-fixing ability. They possess NIF genes. Blue-green algae, cyanobacteria, are of two types: nitrogen-fixing biofertilizers and non-biofertilizers. Nitrogen-fixing biofertilizers are of two types: unicellular, heterocyst absent, and multicellular filamentous. Multicellular filamentous are of two types: heterocyst present, for example nostoc, Anabaena, etc., and heterocyst absent, Oscillatoria, spirulina, etc. Characteristics of blue-green algae: photosynthetic and autotrophic. Nitrogenase enzyme present for biological nitrogen fixation. Non-nodule forming biofertilizer. Heterocyst, nitrogen fixing ability. Specialized cell. It is an enlarged vegetative cell with transparent cytoplasm surrounded by a thick wall internal to the original cell wall. Intercalary or terminal in position. Contains genes for nitrogen fixation. Devoid of photosynthetic pigments. Release vitamin B12. Auxins induce growth of higher plants. Azolla, Anabaena symbiosis. Azolla. It is an aquatic fern freely floating hydrophyte in stagnant water. Air spaces in the leaves of Azolla are occupied by a cyanobacterium Anabaena azole. Anabaena multiplies in the air spaces and remains there till the decay of the leaves. Anabaena fixes nitrogen from the air into nitrogenous compounds which accumulate in the air spaces in the leaves. They are released in water after the decomposition of the leaves. Azolla is a very useful biofertilizer for paddy fields. Medicinal plants. Adhatoda zeylanica medic, Vazaka. Morphology of the plant. The plant is an evergreen perennial shrub. The leaves are large, opposite, lanceolate and dark green in color. Parts used in medicines are roots, leaves, flowers and fruits. Active principle. Alkaloids, for example, vasicolin, adhatotic acid and vasiocolinone. Volatile oil, example, vazacin and betaine. Medicinal uses. Bronchitis, expectorant, febrifuge property, skin diseases, rheumatic joints, insecticides, cold cough and asthma, packing or storing of fruits, blood circulation, tuberculosis. Aloe vera. Morphology of the plant. Aloe is a perennial, succulent, juice-storing, xerophytic herb. Its leaves are large, sessile, crowded, lanceolate, pale green, fleshy with spiny margins. Parts used in medicine, leaves. Active principle, barbaloin, a crystalline glycoside, isobarbaloin, glycoside, anthraquinones, aloamidine, collectively known as aloin. Medicinal uses, piles and rectal fissures, local inflammations, ophthalmia, spleen diseases, liver disorders, hair tonic, antihelminthic, cosmetic. Ocimum sanctum. Morphology of the plant. It is a much-branched erect herb. It is a well-known sacred plant of the Indians. Stem is quadrangular, hairy, green or purplish. Leaves are simple, opposite with entire or toothed margins, gland-dotted and aromatic. Fruits are small and seeds are yellow or reddish in color. Parts used in medicines, roots, leaves and seeds. Active principle, garvacol, eugenol, caryophyllene, alkaloids and tannins. Medicinal uses, insecticides, earache, skin diseases, common colds, urinogenital complaints and malarial fever. Asparagus racemosus, Shatavari. Morphology of the plant. The plant is a spiny xerophytic shrub. The roots are adventitious, store reserve food and form root tubers in fascicles, bundles. The leaves are spiny and modified into scales. The branches are modified into cladodes. Each cladode is a leaf-like needle-shaped and fleshy structure of 1 internode length. Parts used in medicines, roots. Active principle, 1. Steroidal saponin, glycosides, Shatavari 1-4, flavonoids and asparagine. Medicinal uses, diarrhea and dysentery, leprosy, tonic, heart diseases, boils and tumors, diuretic, appetizer, abortions and delivery. Azadirachta indica. Morphology of the plant. It is a perennial evergreen large tree with a blackish bark. Stem is erect, branched, woody with characteristic smell. Leaves are alternate, bipinnately compound and leaflets have a serrate margin. Parts used in medicines, root, stem, bark, leaves, flowers, fruits and seeds. Active principle, in bark, penta, nortriterpene and nimbin. In leaf, quercetin and B-cytosterol glucoside. In seed, azadirachtin. Medicinal uses, skin diseases, intestinal worms, urinary diseases, malarial fevers, demulcent tonic, insecticide, inflammation, jaundice. Mushroom cultivation. The mushrooms belong to the group fungi. They are heterotrophs, saprophytes. Mushrooms are the members of the class Basidiomycetes, for example, Agaricus, Auricularia, etc., and Ascomycetes, Morchella, tuber, etc. Structure of a typical mushroom. Umbrella-like structure, basidiocarp. It is a fruiting body, fructification of the fungus. Basidiocarp produces non-motile basidiospores. It has a very little underground cottony mycelium. The basidiocarp consists of 2 parts, stipe and pileus. Stipe is the basal sterile stalk-like structure and pileus is a terminal enlarged fertile part of the fruiting body. Types of mushrooms. Edible mushrooms and non-edible mushrooms. Edible mushrooms are of 2 types, gill mushrooms and non-gill mushrooms. Gill mushrooms examples, white button mushroom, paddy straw mushroom. Examples of non-gill mushrooms are puffballs and geestrum. Non-edible mushrooms are of 2 types, poisonous and non-poisonous. Poisonous mushrooms example, Amanita muscaria. Non-poisonous, Majority of the non-edible mushrooms are non-poisonous. Temperature, moisture, ventilation, good spawn are required for mushroom cultivation. Steps involved in mushroom cultivation. Obtaining pure culture. The pure culture of mushrooms is obtained from a national or international mushroom culture center, or also can be obtained by isolating the mushrooms from nature. Spawn. Mushroom mycelium coated on some sterilized base represents spawn. Preparation of composts. Compost is a substrate on which mushrooms grow. For better mushroom growth, a mixture of wheat straw or paddy straw plus many organic and inorganic fertilizers is preferred. Such compost is rich in cellulose. Animal droppings are also mixed with the compost. The material is piled in a heap and allowed to compost for about 4 weeks. Casing. The process of covering the layer beds with a layer of soil or soil-like materials is known as casing. It gives support to mushrooms and maintains temperature and prevents drying of the compost. Casing is essential for inducing the fungus to form the fruiting bodies. Spawning. The process of introduction of spawns, fungal mycelium, into the compost beds is called spawning. Harvesting. Mature mushrooms are picked up without disturbing the neighboring ones. During this period also, environmental conditions are left undisturbed. Harvested mushrooms are dried and packed into polythene bags for marketing purpose. They are stored in cold storage godowns.